Welcome to the latest instalment in our Linnean Lens series of online lectures. My name is Wilbur Harrell. I'm the librarian here at the Linnean Society. And it's my very great pleasure to welcome Dr. Isabelle Charmantier, our head of collections, and Ollie Crimmen, our honorary curator of fish and shells. And today they're going to be talking about some of the I hesitate to say beautiful specimens of dried fish, but certainly fascinating specimens of dried fish from our collections here in the Society. Just a little bit of housekeeping before we get started. Firstly, I'm sure you're all dab hands at this by now, but we'd be very grateful if you could mute yourselves and turn off your webcams while Isabelle and Ollie are speaking. And secondly, please do feel free to pose any questions that might come to you during the course of the presentation. We'll have a bit of time at the end, I hope, to tackle some of those. But it would help us tremendously if you could post any questions into the Q&A box at the bottom of your Zoom window rather than the chat. This will help me keep track of the questions as they come in, and hopefully we can avoid missing anybody out at the end. So, without further ado, over to Isabelle and Ollie.
Thank you. Thank you, Will. So today we're going to talk about Alexander's fishes in the Linnean collections. And the way we are going to articulate this Linnean lens is I'll give a short introduction about Garden using a PowerPoint, and then I'll pass over to Ollie, who will present some of the specimens. We have a special document viewer, which allows you to see the fish almost as if you were there, hopefully. So it's a mixture of PowerPoint and another extra video. So hopefully between all this technology, we won't get confused and everything will go well. Okay, so just a few words about Alexander Garden and his fishes. I'm going to share the PowerPoint. There we go. I hope everyone can see it. So the collections of fishes in the Linnean collections comprise 168 specimens. And the major part of these was sent by Alexander Garden, seen here on the left, who was a Scottish physician, mostly interested in botany first and foremost. Garden left Scotland for Charleston in South Carolina in 1752 at the age of 32, and he remained there for 30 years. And because he was actively a physician, so he dealt with his everyday job was to cure and heal people, but he was also very much interested in natural history. And as he wrote to the merchant, the cloth merchant, who was based in London, John Ellis, he wrote to him, "There is not a living soul who knows the least iota of natural history in South Carolina." So all of his conversation about natural history had to happen through letters that he sent mostly to London and after a while to Linnaeus. His network included the cloth merchant, John Ellis, the botanist, Peter Collinson, John Bartram in Philadelphia. And it's through John Ellis that Garden attempted to make contact with Linnaeus. And it took several attempts before the Swedish naturalist finally wrote back in 1759, but instead of wanting information on plants, what Linnaeus wanted from Garden was for him to turn his attention more to zoology and especially to fishes. So in his response to Linnaeus on the 2nd of January 1760, Garden wrote, "Before the receipt of your letter, I had scarcely paid any attention to our fishes, but all your wishes are commands to me, and therefore I immediately set about procuring all the kind I can." So Garden embarked on collecting insects, snakes, even a turtle, but mostly fishes for Linnaeus. We know through research that's been undertaken by previous collector, most notably a curator called Alwyn Wheeler, that he sent 6 consignments to Linnaeus between about approximately 10 years. And at the same time, he was busy as a physician in Charleston. He even dealt with a severe epidemic of smallpox. So these consignments are known because Garden would send letters containing numbered lists of specimens and describing them according to the Linnean method. And he sent these letters to Ellis, who would then, to John Ellis in London, who would then transfer them to Linnaeus in Uppsala. And the letters are full of the practicalities of collecting, naming, numbering, and packing specimen, and their shipping in a time of war between England and France. And in fact, there's probably one consignment to Linnaeus that never made it to England because the ship must have been caught in this war. So for instance, we know that, Arwen Wheeler's study of the Linnean fish used the correspondence between Garden, Ellis and Linnaeus to determine the amount of time each consignment took, from Garden preparing it to Linnaeus actually receiving it. And in 1760, for example, it took about 8 months between the moment that Garden prepared the specimens to the moment that Linnaeus received it. So, for instance, he had collected enough fish to send to Linnaeus by January 1760, but he writes to Ellis, "I would have sent Linnaeus's letter, at least one copy of it, by this opportunity, but as I have not got some of the fishes sufficiently dry, I could not pack them without running the risk of losing them." And then Garden was too preoccupied by an epidemic of smallpox to do much about it until April, when he finally sent the consignment to Ellis in London, who then received it in June and then sent them to Linnaeus, forwarded them to Linnaeus, who probably received them by the end of the summer. So it's quite a lengthy process and a risky one because not all the consignments were received. Almost all the letters were not sent directly to Linnaeus for some reason, even when Garden was in contact with him, but through John Ellis in London. And then in this way, the same specimen could be used and researched twice in many cases, because Ellis would also have a look at the specimen. But Garden was clearly concerned about the effect this would have on the fish. And he writes to Ellis in April 1761, "I should be glad that you had an opportunity of seeing them, but I'm afraid that packing and unpacking them will hurt them." So this material, these fishes are important because they represent really some of the earliest surviving material from North America. And Linnaeus studied and incorporated them into the 12th edition of Systema Naturae, 1766, and in the Mantissa Plantarum as well in 1771. So as a result, quite a few of them are type specimens. That is, they're really the voucher that goes with Linnaeus's name. And for Garden and Linnaeus, I think, the use of specimens as opposed to illustrations were essential to determine the classification of the fish according to the Linnean method of classification, which included using the number of fins and number of scales on a fish. And time and again, Garden refers in his letters to the Linnaeus method of writing characters. And that's really the very formalized way Linnaeus had of describing organisms and especially plants, but which Garden tried to emulate when describing fish. And you can see here on the screen one of the lists that Garden sent along with his consignment. So it's a list of all the fishes. And as Ollie will show you, the numbers that are assigned to each fish are also on the fish. So the number is really the linchpin between the list and the actual specimen. And there's a few interesting things. So most of the list here is in Garden's handwriting, but you can see that Linnaeus has made his own annotations on the list. That's Linnaeus's handwriting. Labrousse is also in Linnaeus's handwriting. So it's very much a collaborative effort of Garden sending the specimen and the preliminary description, sometimes even an indication of what the genus and species might be, and then Linnaeus taking that information and verifying it before printing it in his works. And in his first letter of 1760, Garden writes that with the skins that he sends, Linnaeus will be able to see the true situations of the fins. This will be more satisfactory to you than a bare, perhaps inaccurate description of mine. Nevertheless, I will subjoin their characters, however imperfectly described. And a further letter to Linnaeus in 1763 indicates that Garden preferred to draw up these characters from fresh fish rather than the dried ones he sent to Linnaeus. He says, as my characters of that fish were then made out from a dried specimen, I have on that account now sent to you characters taken from a fish newly catched from the sea, and as I think them much fuller and exacter, I shall beg that you will only pay regard to them. So there's a use of the fresh fish for the description that is sent along with the dried specimen. So that's a bit of context about who Garden was and what his relationship with Linnaeus was and how he sent the material to Linnaeus. And I will pass now, I'll stop sharing and I'll pass over to Ollie to show you some actual specimens.
Good afternoon. So yes, those specimens dried, that's a way of stabilizing biological material, and it's stood the test of time. These fishes are over 200 years old. And so Garden would have been aware as a member of the Royal Society of the methodology that had been expounded by Gronovius to the Royal Society of preparing fishes dry. Herbarium sheets for plants was an established method. With a fish, you've got something more three-dimensional, and it was necessary to gut the fish and clean the insides from the skin, and then the fish could either be kept whole by stuffing it or cutting it in half and sticking it to a sheet of paper, just like a herbarium specimen. This would have enabled an investigator to see things like the number of fin rays and the arrangement of the scales, which are features which are used in the description. These fishes, remember, that Linnaeus is using as a foundation for the modern study of animals, and we're using the same method and in many cases the same names still today. So in this foundation study, these features, these very important features, must be preserved in the specimens so everyone can verify them. So yes, Gronovius's method of drying the fish out, and he would then send those skins to Linnaeus, and Linnaeus would mount them onto sheets pretty much like herbarium sheets. Later, some of them were transferred to cardboard backing, and as we shall see, quite important to keep them flat. So let's look at some of the specimens. Here we have a shark sucker or a mora. And this is Garden's specimen. As Isabelle remarked, Garden numbered them, and you can see the number on the tail label here. That's Garden's own label. The important principle of numbering specimens and tying all the data to it via that number is actually exactly what we do today with museum registration numbers. The details that Garden took from fresh specimens or from freshly acquired specimens, and that he was able to supply to Linnaeus, really it contributed to this modern approach of getting the important details attached to a specimen that can be re-examined. So if we look at this dried out fish, this is a sort of almost a compromise between a whole fish and a half skin stuck to a sheet. So most of the fish is there, and you can see very distinctly the looks like the sole of a shoe, this sucker on the head of the fish, and that is the device by which these animals attached to turtles, boats, even large cetaceans like whales. They get a free ride and they get food as the host eats. They're often found attached to sharks as well, sharks and rays. And that organ is an adapted dorsal fin, so that it looks like a set of Venetian blinds that can be moved to create a suction cup. So it's a successful method. The other way, of course, was to preserve a whole body in alcohol. And that's a lot more hazardous. Alcohol, you can get knocked on the head for your alcohol in certain parts of the world. And some of Linnaeus's collectors, because he had collectors going around the world, were in danger of that problem. But you do then get a whole specimen. The Swedish museum does have some Linnaeus specimens in liquid. And this is a shark sucker, and you can see that the dried specimen has preserved the feature that we like to see, that sucker on the head, and you've got the animal in 3 dimensions. But most of the details that Garden provided were used by Linnaeus in the description, and they're still the features that we use to delineate these, to classify these animals today. That, by the way, is the way the fish swims, of course, with the sucker on top, giving it a very flattened head.
Ollie, do you want to say where this specimen is from? Because it's not part of the Linnean collections, the one you've just shown.
Oh, yes, sorry. Linnaeus's specimen from Garden, of course, is that dried specimen we've seen. This is actually from the Natural History Museum collections, and collected much later. We have dried specimens in the museum as well. There are advantages and disadvantages of the dried specimens. Ironically, some of the fluid preserved specimens have been, since 1900 anyway, preserved in formalin or pickled in formalin, and that wrecks DNA. In actual fact, these dried specimens give very good DNA yields because drying has preserved the molecular tissues. So that's a shark sucker from Garden, and very exotic at the time. And here we've got a slightly more familiar kind of fish. Just switch over cameras. A seahorse. And 2 seahorses, in fact, and Linnaeus became the first person in 1758 to describe a seahorse, naming it Signathus hippocampus. And these 2 specimens that were sent by Garden were used definitely in the description. It could be that between the 10th and 12th editions, Linnaeus actually altered his description a little with additions to account for the second specimen. What we've got here, unfortunately, is 2 different species of seahorse, and there are arguments about which of them Linnaeus was using in his description. Linnaeus also mistakenly thought that the smaller of these 2 was the female and the larger the male, but we can see in the smaller specimen the remains of a brood pouch, because one of the charming features of these little sea fishes is that the female transfers eggs to the male's brood pouch, and he does all the sort of incubation of the eggs, and that's in a swollen pouch in the belly. So it's the little one here that's the female, but they are actually 2 different species. And whilst the taxonomic confusion continues to this day, as people try to work out which animals Linnaeus was definitely talking about, these constitute potentially type specimens, and they remain very important in the study of seahorses. We've also got a ladyfish, Elopsaurus. Thank you. Evidently, Garden had a bit of a problem with this specimen. It looks like he's had to cut it in half to fit it on the sheet. Still, all the salient features are preserved. What's interesting about this specimen is that it's got no scales, because as Garden wrote to Linnaeus, number 15 is the only specimen of this fish that I ever saw, and the gentleman who was kind enough to let me have it had unluckily ordered it to be dressed for supper, so that the scales were taken off before he thought of me, and hence you'll observe that I could not see the natural appearance of the fish or make the characters complete. So somebody had already scraped off the scales getting ready to eat this thing, and then had thought, perhaps Garden would like to see this. And indeed he did. This actually still happens today when we're exploring for new species of fish new to Western science. In tropical regions, we more than once have had to ask somebody if we could have the rest of their supper, because it looks like they're eating a new species. And I think that's actually quite an important point, that these are new to the modern academic study. I say modern, Linnaeus is founding this modern study, as we say. And the fact that people are eating them in some place in the world means that it really has to be a collaborative effort if it's going to be at all successful. And we always work with local people who know the local fauna. And even if they're eating it at the time, we may be able to find new species. So the next fish that I've got here is, so if I missed one out, have we got the rest somewhere? Oh yeah, sorry, yeah, I've gone in the wrong order, sorry. So here's a mullet, a grey mullet. Now this species had been described by Catesby, and it's very interesting to look at Garden writing to Linnaeus about this specimen, because he goes at length to show that this specimen of his shows features that are not available in Catesby's illustrations. So he actually writes to Linnaeus in January 1760, talking of the inaccuracy and inability of this writer, Catesby, in describing and delineating natural objects. Please to observe the albula, our mullet, and you will immediately perceive that he has not only forgotten to count and express the rays of the fins, but that he has, which is hardly credible, left out the pectoral fins entirely and overlooked one of the ventral ones. And not satisfied with that, he delivers a final blow. It's sufficiently evident that his sole object was to make showy figures of the productions of nature rather than to give correct and accurate representations. This is rather to invent than to describe. So he's really laying it on thick there. And I won't say that amongst some of our more egotistical modern investigators who become very bound up and focused on their subject, that this sort of strident criticism does still happen today. But yes, Catesby had, by omitting fins, rather not done this fish justice.
So Ollie, I actually have an image of the fish in Catesby. Shall I? I'm going to share my screen very quickly so that.
Well, this is the mullet. I think we've got. You might have the wrasse, or
I've got both. I do have.
Let's have a look at the mullet. Yeah.
Yeah. So there it is.
Yeah. And you can see the fins missing. Yes, that's excellent.
And I think I may have. Oh.
You can see Garden's got a point, hasn't he?
Yes, yeah, because that fin there has just completely disappeared in Catesby. I thought I had, sorry, I can't seem to be moving my, oh, there you go. That's the, but that's the wrasse that you wanted to go on.
Yes.
To, yeah. So I'll leave it on the PowerPoint while we change the fish.
Yes. So the wrasse you can see there. One thing that you can observe in Catesbury's illustration is some of the colour, and that is actually a notable feature of the wrasses, and it's notable that when you dry fishes or preserve them in alcohol, you lose the colour. This has been a problem throughout the history of modern description. Nowadays we have colour photography that enables us to blast away and take many images in the field, and so we do have the colours easily recorded. But in early times it was important to get colour down recorded at the time the specimen was being first encountered, because subsequently any preservation was going to, as you see, lose all the colour. And it is a very brightly coloured species. If one looks at a, I'll do it on here. Okay. If you look at the top figure there, that's the species in question. And these wrasses you can see are very brightly coloured. And so the colours are used in the descriptions. Wrasses actually have different colours in the males and females and in the juveniles and the adults. So they're a perplexing problem, and their taxonomy has been very confused because of that. So anyway, here's our specimen. In writing to Linnaeus, Garden was actually, he forwarded another wrasse as well. And he says, he describes the fishes as both most beautiful fishes when they're first catched alive. But alas, the luster, glory and beauty of their colours and hues are of short duration and cannot be preserved. Nay, indeed, a great part of their beauty dies with themselves. Well, yes, I think you can say that's true. But the fin rays can be counted, the scales counted, and a species description formulated. So here's a trumpet fish, and looking at the head, you can probably see how it gets its name, with that long sort of tubular mouth parts, or it's a long tubular head. Actually, the jaws are just a small jaws at the end of that tube, and it's a suction feeder, sucking small invertebrates out of crevices in the reef. This one was actually collected by a black servant that Garden sent to the island of Providence, to now part of Colombia, to do do some collecting. Again, modern expeditions still use local help. Very often you get local volunteers caught up and interested in what you're doing, and they offer to go off and collect, and they know environments that will be fruitful. They often know species that one has never seen before. And so this is Garden must have shown him the method of preserving the fish, and he went to Providence and collected this specimen. He collected several fishes, but when on his return voyage, he was in dread of shipwreck. They obviously encountered extremely bad weather, and he had to sort of kind of abandon the specimens to their fate, and many of them perished. But nevertheless, Garden was able to send several of these specimens to Linnaeus. There are less than 10 actually still extant in the collection, and this stuffed specimen of a trumpet fish, referred to as number 11 trumpet fish, is clearly the specimen that was collected by that servant. So yeah, it's intact and clearly describable to the modern species Orlostomus maculatus. So that's a handful of the specimens in Linnaeus's collection. Gives you an idea of the sort of condition. Many of them type specimens. So they still must be referred to in modern studies. We pin our names to individual specimens, of course, using type designation. And so these are not only vital specimens in modern studies, but have this fantastic historical interest going right back to the foundation of modern ichthyology and the first specimens to be seen from the New World.
Thank you, Ollie. I just wanted to add to that all these specimens have been digitized and they're freely available on our online collections, as the plants, the shells, and the insects. So if you want to find out more or just browse to see the fishes, you can find them on our online collections. And also, I'm going to put in the chat 3 very useful pieces of literature on Alexander Garden and also on the Linnean fish collections here at the Linnean Society, in case that's useful. And thank you, Will. You've put in the link to the online collections, specifically the fish. So that's it for our short presentation. If anyone has any questions, either put it in the Q&A, or you can also raise your hand. And I think I believe there's a way of unmuting. We might have to rely on Padma for that. Thanks, Padma. So please don't hesitate to ask any questions to either Ollie or I or Will. I see there is one already in there.
Would you like me to read
sure,
out some of the questions? So the first one is from Rhonda, and she asks, what was Ellis's connection to the group of scientists? I presume that's Garden and possibly Linnaeus.
Yeah, so Ollie, do you want me to take that?
Sure, yes.
So John Ellis was he was a British linen merchant, and he was appointed royal agent for British West Florida in 1764, but also he was a fellow of the Royal Society, and that's why that network that's where the network originated. So Peter Collinson was also a fellow of the Royal Society, and both of them, along with other naturalists, were in correspondence with Linnaeus. So that's where the network comes from. Ellis did quite a lot of work on, well, his most famous work, I think, was on coral and the Venus flytrap as well. So he had he had that kind of interest in the natural world.
David Hibbler asks, do we know anything more about the relationship between Garden and Catesby, aside from their frosty reviews of his work?
Yeah, no, I don't. I don't know if you do, Ollie, but I don't know.
No,
whether they
clearly the Garden had a lot to say about Catesby's work and lack of detail. Linnaeus would have been using Catesby's details, but the network that we're talking about there, it is exchanged in correspondence. A lot of these people didn't meet or communicate in as readily as we can now, of course, and their characters are not, there's not so much information about their characters, so one relies on these letters, and they're not always very detailed about their feelings about each other.
Yeah, and I should say Catesby, actually, I was just checking my dates with Wikipedia. Catesby had died by 1749, so he was already dead by the time Garden arrived in South Carolina.
Oh, right. Okay.
So they could never have met, but I think Catesby was such a monumental work, and as you say, Ollie, you know, used by Linnaeus in his previous works of Systema Naturae, that it would have been it would have been a reference for Garden to use. And I think yes, Garden was clearly not impressed with Catesby. I had yeah, he wrote. So I did find the Alexander Garden letters are absolutely fascinating, I have to say, and they were translated and published by James Edward Smith, who was the founder of the Linnean Society, in this volume of correspondence of Linnaeus and other naturalists. And when I started researching Alexander Garden fishes, I started delving into the letters, but actually they're a fascinating read. They're really they're really easily readable and very entertaining. So, for example, Garden wrote to Ellis in 1760 saying, and I suppose that's from the moment that Linnaeus asked him to look at the fishes. So he writes, "I never before looked to our fishes attentively, but I am struck with astonishment at Catesby's blunders. Sometimes he forgets whole fins, etc. In a word, there is nothing can possibly recommend him but the specious beauty of the coloring of the plates." And then a year later, he writes again saying, "Oh, my good friend, how many blunders and gross misrepresentations have I seen in Catesby, gross beyond conception." So he was clearly quite shocked, actually, by the mistakes in Catesby, which I suppose he would have taken for a good representation of the fishes before he started actually delving deeper into Catesby for fishes for Linnaeus.
I suppose now that as you've pointed out, that it didn't overlap, he's not going to get any comeback, is he?
No. Are there any other questions? As I say, you can raise your hand if that's easier, or put it in the Q&A.
In the meantime, Isabelle, I thought one thing might be worth mentioning, that that drying method of preservation means this specimens are very stable. What we have to do with the Society's collections is protect them from humidity and insect attack. But otherwise, if the conditions are fairly stable, these things should last really for hundreds and hundreds of years. Alcohol specimens are less stable. Of course, you've got the problem with flammability and a fire hazard. But the dried specimens are relatively low maintenance, providing we keep stable conditions, which we do have here at the vault in the Society.
That's true. Don't you have to keep topping up the alcohol?
Pretty much inevitably. I mean, we do try to get the containers that are well sealed to keep the alcohol from evaporating, but no, they do always slowly evaporate. And if they dry out, it's not so good as if they're dried from the fresh specimen, because then you've got some damage because of the way the alcohol has soaked into the tissues.
I see there's another question, actually. Sorry, Will was busy getting the phone. It's Rhonda again. She says, "Can you talk a bit about the dangers of using alcohol in certain parts of the world for the early collectors?"
Well, yes, I did read of some of Linnaeus's collectors who were sent to tropical parts having had their alcohol stolen, and in one case the collector was physically attacked. Alcohol is a sought after commodity all over the world, and it's heavy, difficult to carry around. Container breakage can leave you with no means of preserving your specimen except for drying. So it was in the early 1700s that investigators started using alcohol to preserve the whole body. Human anatomists had invented the technique, really, and in some voyages, notably Captain Cook's voyages around the world, in the 1760s, many of the specimens arrived in alcohol, not unlike the ship's rum. Sometimes It was the ship's rum that was used to preserve whole specimens. But it's ethanol, it's the drinking variety of alcohol, and we do actually use the same liquid in modern collections. The only thing is that it's very often denatured industrial alcohol for which we pay lower duty. But yeah, hazard of fire, theft, and attack for somebody who wants to drink your alcohol have been recorded by several collectors.
Interesting. Actually, it's in some of Garden's letters. He is indicating that there's a whole shipment that he sends. They're in rum.
Yeah. Yeah, it's anything above 50–60% strength is going to act as a preservative.
I've had one more question come in, two more now, in fact. Alice asks, have there been differences noticed between these specimens collected by Garden and the modern fishes today? Well,
yes, and I think the answer there is there you see the value of these specimens, because if the differences are sufficient, then we're talking about a different species. If you're talking about changes in members of the same species over time, then that would be difficult to examine from these specimens, but it could be done. So for instance, if somebody was looking, a modern example, and it's not really going to apply to these specimens, is people would like to know when microplastics first turn up in aquatic environments. And so you look at old specimens and find there were no microplastics ingested by fish. And in modern ones, you nearly always find that sadly they have. But although Linnaeus and Garden's specimens that wouldn't have been an issue, the isotopes, chemical isotopes that are contained in these skins might be of interest. Now, obviously, there you've got a bit of a challenge, as with all preserved museum specimens. You've got to balance how much you might want to put invasive techniques on that specimen. You know, if you're scraping muscle tissue or skin away in order to do a chemical analysis, you've got, especially with these collections, to weigh that up against their historical importance and weigh it up against techniques that we don't know about yet. So our methodologies are evolving all the time. We couldn't have foreseen ourselves looking at DNA or doing CT scanning in previous times. So the curators and the guardians of these collections have to weigh up what can be obtained from modern studies and what's worth sacrificing small bits of tissue for, etc., against the long-term preservation of the specimen. But yes, it may be that examining these skins will give us answers to interesting questions about the environment. It's possible.
Thank you, Ollie. We've got a few more questions come in. Jim asks, well, he says, fascinating session, and then asks, how were the spliced fish dried? Were they pressed or simply dried out in sunlight for a period of time?
I think they were dried out in the sun. And you hear from the correspondence from Garden talking about the fishes not being dry yet. It was obviously done naturally, and not sort of heating specimens is very injurious to try and dry them that way. But sunlight and wind, I suspect, was the way that it was done. So like hanging out the washing, really allowing nature to dry them rather than trying to speed it up in any way, because what you don't want is distortion, and that's another reason why they're prepared flat.
And I think they were also varnished, no?
Yes, for the long-term preservation, they were varnished as much for presentation, I think, as anything else. They didn't really, the varnishes, protect much against insects. In fact, sadly, some of the varnishes seem to attract insect pests, but they certainly make certain features more visible. You can see the outlines of the scales better, and So yes, for presentation purposes, they were very often varnished.
Gina asks, do you have Mary Kingsley's specimens in the NHM collection, and are they in alcohol?
Yes, Mary Kingsley was a Victorian collector who intrepidly travelled across parts of Africa, collecting fishes and reptiles for the curator at the Natural History Museum, and she preserved her specimens in alcohol. This is, of course, a century later than the ones we've been looking at today, but they are still in the museum. And likewise, when Albert Gunter of the museum saw her specimens, he was often able to describe new species on the basis of those specimens, and they are type specimens in the same way that many of the specimens in Linnaeus's collection are types.
And then Rhonda asks very straightforwardly, how are the dried specimens stored?
Well, as long as you don't get humidity into the system, because humidity obviously exposes the specimen again to water, atmospheric moisture, and that can allow the biological degradation processes that drying stops to start up again. So you can and you can get mold growing on the specimens, and they can actually be destroyed subsequently to their preservation if they get wet again. So the trick is to keep them dry, keep them from being exposed to moisture, and then as long as they're kept flat and in that dry, stable environment, well, you can look on these collections that we've been looking at as a long-running, very long-running experiment in how to keep biological specimens, and we hope we can keep them forever. That might be a bit optimistic, but who knows?
Actually, I think that's a lovely way to end.
That's a nice note to end on. So I'll just extend our thanks to Isabelle and to Ollie. That was a wonderful talk. It was fascinating to watch here live. It was a bit like dinner service. Ollie was sat at a desk here in the annex, and the dried fishes were brought to him in the least appetizing meal. It was wonderful to see them. I completely retract my remark about the beauty of the specimens. They were wonderful. It was really lovely to see those. So if there aren't any more questions, I think we will wrap
There is
up.
actually.
Oh, is there? Oh,
yeah.
gosh. Well, we have a bit more time. Rhonda simply says, thank you. A fabulous presentation, and she's tuning in from New York City. And then a secondary question, have there been instances where parasites are dried along with the collection? I suppose that's parasites of the fish, or maybe insect pests that might otherwise have predated upon the specimens.
I don't know, Isabelle, if you've discovered any parasites so far in the Linnean collections, but certainly I do know that with alcohol preserved specimens, definitely you get the parasites preserved from in the insides of the fish. There'll be a lot of nematodes and other parasites included in the guts of the fish and in the muscles, and I suppose that's one advantage of the alcohol preservation, that you do get all that. But a lot of parasites attach themselves to the outside of fishes, so you get copepods. Some copepods actually go into the mouth of fishes and eat away the tongue, and then replace the tongue as a sort of crushing plate for the food, so they remain in the mouths of the fishes. Parasitologists do go through museum specimens looking for parasites, and although I'm not aware of a case yet with the Linnean collections, who knows? They could certainly be there.
Right. Well, lots of people saying thank you for a wonderful talk. Joe and Julie both saying thank you for your speaking to us today, Ollie. I think the only thing extra I had to say was that if you've enjoyed this talk, please do feel free to come to some of the others. We hold them on a bi-monthly basis now, the Linnean Lens series. So please do keep your eyes glued to our events pages for further instalments and to the other events that we have at the Linnean Society, of course. And if you've enjoyed looking at any of the specimens today, you might want to look at other aspects of our collections. We have a new book out called L50, Object Stories and Discoveries from the Linnean Society, highly reasonably priced and a very good read. And I think someone has put a link to our bookshop in the chat. And on that note, I think we will say thank you very much and see you again.
Thank you. Bye-bye.